What we are going to discuss today, this paper is a great paper that was published in Nature Immunology. Program disarming of the neutrophil proteome reduces the magnitude of inflammation. These today are, how you already know, we have the presence of the first author, José Ma Adrover, and the corresponding author of this work, Andrés Hidalgo. And well, to recapitulate the knowledge about the basic knowledge about the cells that are studied in this paper, we know that neutrophils are an important innate immune cells, are polymononuclear leukocytes by the segmentation of its nucleus. These cells are important in acute inflammation responses against bacteria, against different pathogen challenges, pathological challenges, and these cells are developed in bone marrow like any other myeloid cells. These cells are developed from myeloid precursors, and these cells, when they are in an ready stadium, they go out from the bone marrow and are released to the blood circulation, where it has a media life of approximately 8 hours in humans. And the neutrophils have diverse effector functions, but the principal can be categorized in 3 different responses. One is phagocytosis of pathogens. These cells can induce the phagocytation of different pathogens to internalize them, and by this way make the improve the degradation of the components of the pathogen, like bacteria. Then there is also the granulation factor function, that is the externalization of the granulates from the exterior to kill the exterior of the cell, to kill pathogens. And also third mechanism is by nets. Nets are neutrophil extracellular traps, and we are going to talk more about it. Granules can be divided in primary, secondary, tertiary, and secretory vesicles by its content. The primary has a xerophilic, are xerophilic in tissue analysis, and are characterized by the presence of myeloid peroxidase. The secondary granules have principally lactoferrin. The tertiary is characterized by gelatinized content. And well, secretory vesicles varies in its content, but all of these granules are vesicles that are released to the exterior and release different cargo that permits to kill another pathogen like bacteria, and also to induce the cytotoxicity. NET is the neutrophil extracellular traps that are formed by chromatin, DNA, and histones that suffer modifications that permit the condensation of this chromatin. Also, it has more proteins like elastase and myeloid peroxidase from the granules that enters the nucleus space and are projected with the DNA. This process can produce the death of the cell, because all the DNA is ejected, it also makes that the content of the cytoplasm could be ejected. And this is a this mode of cell death is known as netosis. But also there is another recent discovered mode of type of nets that is for mitochondrial DNA, and well, this is also interesting because not necessarily the cell death is that it can be alive after the ejection, mitochondrial DNA ejection. And well, the purpose of nets principally are to reduce the mobility of pathogens and to promote the death of the different targets, pathological targets, by the releasing of the different proteins, like proteins that degrade also the extracellular matrix that permits the recruitation of more immunological cells to improve the response against the pathogen. And well, the molecular circadian clock, for the another side, is an important concept that we are going to abord in this, that the authors abord in this work. And the molecular circadian clock are present in many of the cells of the body, and it consists on different proteins that have a circadian variation of its expression, and forms loops of regulation. We have clock and Bmall that regulates the expression of different clock genes, including PER and CRY, inducing the expression of these, augmented its presence of cytoplasm of these proteins. These proteins then inhibits the Bmall and clock, and by this way they inhibits its own concentration in the cell. And by this, in these loops of regulation have a 1 day duration, so it repeats every day. And the importance of this is that not only is these genes involved, but there are more genes that are involved in this regulation, and these more genes also regulate the expression on many other genes. So this is known as the molecular circadian clock, and it's present in different tissues. But it's important to say that in the brain there is a suprachiasmatic nucleus that is named the central molecular clock, and also in the different tissues like liver, like intestine, like in other tissues, and immune tissue like neutrophils have peripheral clocks, by including parts of deep machinery, but also regulating different sets of genes. And well, it is known that that there could be communication between the different molecular clocks. And well, to introduce to this work, the last paper from José Adrover, before this, the present work, is this where they discovered that the activity of neutrophils is regulated in a circadian fashion. They discovered that neutrophils can be divided or classified by fresh neutrophils and aged neutrophils. It is important because it infers a process that aging in the day in the lifetime of the neutrophils, and this process they discovered that is regulated by Bmall, is implicated, a participant of molecular clock that regulates the expression of CXCR2. CXCR2 in an autocrine manner activates the CXCR2 receptor, and this receptor in the neutrophil promotes a signaling cascade that induces the aging phenotype in these neutrophils. And well, they discovered by how this works, and they produce Bmall knockout mice and CXR4 knockout mice specific both in neutrophils, and they saw that you can have constitutively fresh neutrophils or constitutive aged neutrophils by these mutations. And also one important observation of this work is that constitutively aged neutrophils cause accumulation of this type of neutrophils on the microvasculature, and this accumulation can induce damage in this tissue because the neutrophils that are here it could induce the granulation and net formation and all these effector responses of neutrophils present every day in this microvascular and accumulated, and the accumulation of this type of responses could induce a considerable injury in this tissue. But for another side, we know that the physiological lifetime of neutrophils is there are developed on bone marrow, and is say that in the night bone marrow neutrophils from madurated from bone marrow are released to the circulation, and it is where they have, as I say, 8 hours of approximately of lifetime, and then they move to different tissues, principally in homeostasis and a homeostasis condition. They can be present in liver, in lungs, and also in spleen. But these tissues, especially the lung, is a tissue highly vascularized. Then how the neutrophils present in the lungs and in other tissues that are highly vascularized prevent the damage, the injury, the vascular damage in tissues caused by neutrophils. And then this question conducted to is there a specific mechanism that prevent the neutrophil inflicted toxicity in tissues? And well, they work in this from this question, and they first want to study if there is differences of the neutrophils that are present in the day, that neutrophils that are in the night in the organism. So to study this, if these populations could be different, they study the proteome of these cells. They want to analyze if there is a proteomic differences between these two type of neutrophils. To obtain a significant protein number, they use a model where they enrich the night-like neutrophils and the day-like neutrophils. The night-like neutrophils are enriched by the administration of AMD 3100, that is an antagonist of CXCR2 that are known to by this way enrich the night-like neutrophil in the blood circulation, and by day-like neutrophils they enrich this by endothelial selectinase antibody administration to these mice, and then they obtain circulated neutrophils from these mice and analyzed by tandem mass spectrometry the proteome of these cells, and they well, in the figure in the panel A we can see that proteins that are differentially present in the different population, night-like neutrophils and day-like neutrophils, and the tags are collocated to proteins that have less than FDR of 0.01. And well, we can say that there is enrichment in cytotoxicity, metalloproteases, vesicles, and different of these set of type of proteins are more enriched in the night-like neutrophils versus the day-like neutrophils. And they make a validation of some of these proteins by flow cytometry to analyze if there is a difference or could be observed by this another methodology, and they observe the same results. And well, they want to know if the proteome differentially present in these two type of population are involved in a specific cellular process, and they do gene ontology analysis of this proteome, and they we can see in red the proteome of daylight neutrophils, and in blue the data from nightlight neutrophils. And this is that's the size of the circle is proportional to how many the proteome data set of these biological of these different groups are overlapping with the specific gene ontology term, like protein metabolism, RNA metabolism, and etc.
From this figure, I was wondering why most of the Go terms that are enriched are in the day category instead of the night category. So in general, the night category proteins that are enriched are also have like less significant p-value. So I'm wondering if there's one enrichment towards day. I don't know if the authors like maybe this is based on the way the experiment was designed, or.
Jose, you want to respond on the analysis? Okay, I'll start with this. So this is more qualitative than quantitative. What we wanted to see here is inspecting this with the omics data. Like everything, this is very much guided for what you're looking for. You will get hundreds, literally, of pathways that are up or down. The trick is always how you select what may be meaningful. So what we wanted to make here is sure that there's more significance on the daylight, and Jose may comment on this. What we care so much is not on the p-values. Hello?
Yep.
Can you hear me?
Yeah, we can listen.
Yeah. So rather than the quality, so what we were more interested in, and the reason why we included this figure, is that the pathways were already telling us something about the type of protein composition that the cells have. So if you look, those at nighttime have some pathways that we thought were interesting because they refer to Wnt signaling, c-KIT signaling, GTPs, things that are typical of bone marrow cells that are forming and are moving actively. If you look at the red spots, is clearly more of immune response, inflammatory responses against viruses, TLR4 activation, and so on and so forth. So this was the main point. Statistics with this typically requires either many samples or a lot of depth, which we were not interested in, basically because we couldn't get a lot of cells. The number of samples here is limited. This is based on many cells, few samples, and that probably also weighs on the p-value. Jose, I don't know if you want to say something else.
Yeah, so just this is one thing to note on this kind of analysis. This is not Go terms. This is GSEA. And here what you see, so when you see one of these gene sets that is that you see on the age side or on the fresh side. So when you see a pathway that is enriched in the aged side, it also means that it's downregulated in the fresh side. So both populations are analyzed at the same time. I don't know if I'm explaining myself.
Yeah. What I'm saying is that I know that in this case, I guess you did like this hypergeometric test in which a set of genes is compared against another set, and then you compare how significantly enriched one set is in another set of genes, basically.
Mm-hmm.
So what I'm wondering is why the day sample gives more specificity than the night.
Well, that's what I was trying to say. So when you see that something is on the day side on the day sample, it also means that it's down on the fresh sample. I don't know if.
Oh, okay, I get it. Okay, yeah. That's true.
Okay, thank you. Then they want to know if this proteomic data set is this type profile of proteins present in this different population of neutrophils have a relationship with the transcriptome, and then for this they compares the proteomic data set to the set of RNA sequencing data, and they show that there is no correlation between these two data sets, and then this is indicative that the proteome variation between these two populations are not by change in transcriptional activity in these cells, and it is logic because the majority of the transcription is patterns are established at the bone marrow, but interesting and also specifically the proteins involved that are present in granules and or the proteins that are necessary for net formation. And this information is interesting. Then they evaluate if there is a differential enrichment in the proteins that are typically present in granules, and we can say here different proteins. Black dots are indicative of proteins from this type, and also the color coded dots. But there is these color coded dots have a differential, significantly differential presence in these two types of neutrophils. They are present in neutrophilic granules, like the pink dots, and the different granules, and we can see that there is a more enrichment of these proteins in the night-like neutrophils, clearly, against the day-like neutrophils. And well, they also want to analyze if these differences in proteome data between these two type of neutrophils.
Something related with the transcription and proteomic part. I think it's very interesting in the previous slide. I was wondering if the authors tried to go a bit deeper into this. Like, I guess there must have been some genes that made a good correlate between proteomic and transcription, and I'm wondering if that subset can explain some of the, like, if that explains if there's a subset that is linked both transcriptional and at proteomic level that can explain a bit this phenotype of dyed enrichment of different granular proteins.
I'm not sure I understand the question very well.
Like the link between transcription and proteomics in the two data sets. Is there a subset of genes that correlates, or there's not a single?
There's a very low proportion of genes and proteins that correlate. So we didn't really look much into detail into that. So I can't tell you really if some of the variation that we see in the proteomics may be explained by like the few genes that correlate. I really can't tell you.
Okay.
I mean, it's a good point, and we thought this was interesting. This was a side result that we thought it was interesting enough to add, but like Jose mentioned, nothing we added in detail, basically, because you do what you can more than what you want. We didn't have, which I would have liked to have, a control population, just to show that this is a thing of neutrophils and not, for example, of monocytes or lymphocytes that are more actively transcribing and might show a completely different curve. That would have been very nice, but of course very involving because we don't need to go into those. So, but the thought behind this is quite, if it's not seen in other cell types, which we did not analyze here. It's very interesting because basically it's telling you that the mature neutrophil in blood basically flies solo without, it does transcription when it needs to, but in normal conditions, it goes with what it has, and that's what we thought was interesting in this case. Also allowed to support the other data.
Good. Thank you very much.
Thank you.
Yeah, maybe
And
it would be interesting to inhibit transcription on these cells and see if you can keep the same phenotype, right?
Yeah, yeah. Well, the problem with neutrophils is that there's a lot of things that you cannot do
Yeah,
because you
that's true.
cannot culture them. So that makes a problem. And if you treat a mouse with a transcription inhibitor, that's probably bad news also. So yeah, we were a bit limited, but that would have been nice.
Thank you. Well, they analyzed the presence of granules in these neutrophils at different daytimes, and they isolated lysis positive neutrophils from blood, and they evaluated the presence of myeloperoxidase that is present, as I said, in the xerophilic or primary granules. And well, we can see that in the night, like and CXCR2 and CXCR2 that are indicatives of site gather time. This is a nomenclature for principally used in the circadian field, and CXCR2 is one from the signs the lights are on the day cycle of the mice, and these hours, the 13, 17, and 21 are hours of the night, and 1, 5, and 9 are day hours or light, or hours with light. And we can see that the granules are more present in the daylight, in the neutrophils isolated at 9, that granule that isolated in the day, and we can see better this variation in this graph, and we can see a typical circadian oscillation of the presence of these granules in the neutrophils. And also they evaluated the concentration of elastase, neutrophil elastase in plasma, and they show in the plasma blood, and they show that there is an increase on a peak on the concentration of this elastase of the day, and there is a valley in the night. And this is interesting because it says that we can see that the neutrophils degranulates along the day, and in the day they have less granules because maybe they are degranulating in the blood circulation, and this indeed correlates that with the presence of neutrophil elastase in the plasma. That it supports the hypothesis that the neutrophils are degranulating in the blood circulation, and this is very clear here. And then they want to know is the neutrophils present in different tissues have more or less granules, and they saw that indeed the lung, spleen, and liver neutrophils that are tissues that are infiltrated from have a neutrophils infiltration in an steady state. They saw that despite the neutrophils infiltrates to these tissues, these neutrophils have no the same quantity of granules like the blood neutrophil cells, and this is important because it is indicative from the question that I say at the start of this presentation, that why the neutrophil infiltration, homeostatic neutrophil infiltration of these tissues are not causing vascular damage, and maybe it's because they are degranulated when they arrive here. So far, we know that there is some differences in the daylight and nighttime neutrophils protein enrichment profile, and that it also can be viewed in the granule proteins that are more enriched in the nighttime neutrophils. And also for granules, we can the authors see that neutrophils from circulation has less MPO granules at the daytime, and the neutrophils that arrive at the tissue have less of these granules compared with blood circulating neutrophils. And this is assuming this phrase that circulating neutrophils degranulate in the steady state in the circulation. So,
Dr. Hidalgo, I was wondering, or Jose, if it's required to degranulate before entering the tissue. So if you block the granulation, do we still have infiltration in the tissues?
Yeah, that's a good question. So we haven't done that in this paper, but in the previous one, you will see that we show in this paper that this degranulation is controlled by the molecular clock. And in the previous work that we did, we analyzed the migration of neutrophils, which are always fresh or always aged. And what I can tell you is that fresh neutrophils, the ones that have lots of granules and are freely released from the bone marrow, these are less efficient migrating to the tissues in the steady state.
okay.
Thank you.
Yeah, I also was wondering about this steady state degranulation and how similar or how different it is compared to a degranulation activated by like encountering a pathogen. Like if it's something that might be just through like a passive process, or its neutrophils are actively degranulating through their lives.
Yeah, that's a good point. The answer is we don't know, because we never compare. I guess an interesting experiment would have been to do a proteome analysis of this type of degranulated neutrophils from blood and neutrophils that we treated with an agonist. Which agonist? It probably depends on the agonist that you get a different composition, and that's it. That's if you don't induce nets, in which case things might be a bit different. So, so the answer is we don't know. What we presume is that this is more controlled, slow degranulation. When you hit with an agonist, you get the granulation changes in the plasma proteins within minutes, very few minutes. And this seems to be taking hours. So the only thing that I would argue is that this is slower. At the molecular level, we don't know. Did not look.
Thank you.
So thank you for the questions. And well, they want to evaluate this effect of differential neutrophil responses in day and night, also in the net formation. So they first evaluated the data of the proteome analysis, and they saw that the night neutrophils, the night-like neutrophils, have enrichment of the proteins necessary for net formation against the day-like neutrophils, and also they evaluated the net forming capacity of these different neutrophils. So they isolated neutrophils from blood circulation of mice at CT13 or CT5, and they saw that in ex vivo stimulation with PMA of these neutrophils, there is more net formation of in the daylight neutrophils or the daytime neutrophils at CT13 than the daytime neutrophils. And they also evaluated this phenomena in vivo by they induce tissue damage by ischemia reperfusion in the mice, specifically in the muscle cremaster, and they evaluated the neutrophil, the net formation on this tissue after this process that induces tissue damage and neutrophil reclutation to this site. And we can see that there's more, and indeed there is more net formation at the nighttime than in the daytime. And well, we have the control the neutropenia mice with that for a control of net formation. And then they analyze if these differences of response in the proteome and effector responses of neutrophils can be related to the mechanism that they saw in the last paper, that is molecular clock Bmall affected by CXR2. And for that purpose, they evaluated the differences in granule quantity in these two type of neutrophils by administering CXL2 to certain neutrophils, and they saw that when they put CXL2, that is ligand of CXR2 receptor, they saw that there is a degranulation in these neutrophils. And B, they saw the same, not only in the wild type mice, but they also analyzed what happens if you quit the components of the CXR2 signaling. If you don't have the receptor, yeah, these change in the granulation are not viewed, and also if there is no the CXL2 present in the mice. So then they also evaluate the net forming capacity ex vivo, simulating with PMA and wild type CXR2 specific depleted specifically neutrophils and CXL2 depleted mice, and they saw that there is, as we know, there's less formation of nets in the daytime, but these difference are not viewed if there is no the CXR2 of CXL2. And well, we know that Bmall regulates the circadian expression pattern of CXL2. So they evaluated if Bmall is involved in the nighttime versus daytime neutrophil differences regulation, and they saw that indeed there is less granules present in the when you don't have Bmall, there is no difference between the daytime or nighttime neutrophils granules, and there is the same also the same quantity of granules, and also the differences viewed in nuts forming capacity in this by these neutrophils at a different time are not viewed when you don't have Bmall. And also the pattern of protein enrichment in the wild type conditions between night and daylight neutrophils are abrogated when you don't have Bmall in the neutrophil. So we can see that there is a clear role of Bmall in this circadian regulation. So net formation capacity is diminished in the daytime neutrophil when you compare this with nighttime neutrophils, and CXR2 signaling and Bmall Bmall1 are involved in the daytime neutrophil disarming, evaluated as PO granules and net forming capacity. Both of them are decreased in the daytime neutrophils. So then they have the preference in the neutrophil viewed in the neutrophils across the daytime are have a role in the injury that neutrophil can cause in the different tissues.
Sorry.
They evaluate here.
Sorry.
See? Yeah.
Regarding the previous figure, I have two questions. I
Here?
wonder, and in the previous one, related to this one, I wanted to know if those mice that are deleted in CXCR2 in the neutrophils, they have an evident phenotype related to damage or related. Yes, because those neutrophils cannot degranulate before they enter to the tissues. So they develop some kind of damage or something like that. And I have another question here. They said that this is an autocrine regulation because the same CXCL2 that they produce are captured by those neutrophils and allow them to degranulate before they enter to the tissues. But in the previous figure, they show that the exogenous CXCL2 that they add had an effect in the. Can you go back to the previous one?
This one.
They observe a effect in the low amount of the granules they show per neutrophil. So, and this is an exogenous CXCL2, but they comment that the effect of the granulation before the neutrophils enter to the tissue, it's regulated in an autocrine manner. Can you explain a little bit of that? Why do you observe a effect added exogenous CXCL2?
Okay, so I'll go for the first one, and Jose can go for the. I'll go for the second question, and Jose can go for the first one if he wants. So I'll answer that too. So this experiment in panel A was just to show that CXCL2 induced the granulation. Now, surprise here, we're not the first ones to show that agonists for this induce the granulation. This is exogenous and at a concentration that was relatively high. The levels in plasma of CXCL2 are very low. Actually, they're so low that we had a hard time to detect it. And this is not in this paper; it's for the previous immunity paper. So here is more, again, it's more a qualitative response. CXCL2 is able to induce this response. The levels in plasma might be too low to recapitulate this, but the key question here is that you have CXCL2 presumably produced locally in a way that we don't know. We don't know if it's associated with anything on the membrane of the cell that makes it more efficient for CXCL2 to recognize. But the reason we know this is autocrine and somehow is working and is not systemic, which is what you would think, if it's released by one cell, then it should signal on the neighboring cell. But if we make a mouse where we combine neutrophils that are wild type together with neutrophils that are CXCL2, so they don't produce the chemokine, then the phenotype, the granule, it's another panel that I think Emmanuel didn't show, but it's okay. Then the phenotype of the CXCL2 knockout is very different from the wild type. So the CXCL2 knockout are unable to degranulate, whereas the wild type that are in the same plasma, in the same blood, have less granules. So the conclusion we got from that, without getting to the details of how this might work, is that the signal is produced by one CXCL2 produced by one cell needs to signal in a cell autonomous manner. How does this work? Why is it more efficient? Why is CXCL2 released systemically, or is it that the concentration in plasma is too low, or there's some inhibitor or protease that degrades the chemo? We don't know, but that's all likely to happen, that the signaling on the same cell is much more efficient.
So I was curious about your idea of how it's possible that the neutrophils, like they are sort of slowly degranulating while they're in circulation, but also looking at the confocal imaging, it seems like as if they are slowly falling apart, while in circulation. But the fact is that probably they are still integer or intact, but they are actually losing pieces of granules from their cytoplasm. And I was wondering if, we know if this is controlled by the same machinery that is usually involved in the polarized degranulation that is usually involved, for example, for their antimicrobial activity.
Okay.
Sorry, Federico, I lost part of the question because my connection,
Okay.
either my connection was whatever happened. Jose, I don't know if you follow it and you want to answer.
So you were asking if this way of degranulation is controlled by the same mechanism, right? Yeah. To all the kinds of degranulation. Yeah. Basically, it seems that from I know the focal imaging doesn't show all the neutrophils in this case, but it seems as if the neutrophil itself is like losing pieces of cells, And so what we think they're doing is they're releasing the granules, but not so the cell integrity it's maintained as far as we know. Yeah.
Jose has a very interesting. We had a very interesting project a couple of years ago about what Jose called the microneutros, microneutrophils or whatever. I don't know what the name was, Jose. And the idea is that we see fragments of cells that are circulating. When you look by intraviolent microscopy, you see just chunks of little particles that have all the markers of neutrophils. They're very small. They don't have a nucleus. So that might be one way in which granules are lost. The cell it's a little bit smaller, so it might be losing some of the content. But for the most part, the cell looks quite normal. And that's one of those projects that we don't know if we will ever pursue, but the idea that these released particles, which might contain some of these granules, what the hell are they doing in the bio? I mean, what's with them? So, answering that question. So.
Okay, yeah. Thank you.
Thanks. Well, we continue. Well, continuing with the paper, they evaluated how was the innate. We can want to evaluate the severity of lung injury or driven by these neutrophils at different daytime and with different profile. And for that, they use the ALI system. ALI is acute lung injury that is driven by administration of LPS, and in 24 hours later, they inject intravenous. They do an intravenous injection of anti-H2D, that is an antibody against a haplotype of MHC that is that induces this mixture of administration drives the acute inflammation in the lung tissue, and we can say in panel A that when they induce this ALI process, we can see more nets like viewed by citrullinated histone 3 and MPO peroxidase. And then they want to know, they analyze the kinetics of this the net formation in this lung tissue, and we can see in the panel B that this kinetic, we can see that there is a formation of nets, and they also disappear later. Well, the importance of this is in C, that they show differential net-like structure measures in the neutrophil, that when the ALI is produced at the night and CXCR2, or when ALI is produced at the day and CT5. And well, they use we can see that in the when this damage is this inflammation in the lungs is induced in the night, that night we can see more net-like structures through this after this induction, and is maintained. And when you induce this ALI in the day, we can see less net formation. And well, like as a control, they use chloramidine, that is a PAD4 inhibitor that inhibits net formation. PAD4 is a protein necessary in the process of net formation. Well, we can see better in this graph. So I want to show you this video where the here is showed. Sorry. Here, this is an intravital imaging of lung during LE, and we can see the net formation, the different times of and the day or the night. When the ALI is formed in the day, we are viewing now. And we can see clearly that there is more net formation or more net-like structures when Ali is performed at night. Then, to this propose this, they want to know if this response are if in this response are involved the platelets, and they saw that indeed there is an increasement in the platelets presence after the early induction, but importantly, this augment is similar between the two times, and this indicates that platelets are not involved in the differential response viewed at the day versus the night. And then they also evaluated the edema formation in the lung tissue when they induced Ali, and by computerized tomography, and we can see here the signal of water infiltration or water content in the lungs after the basal base at the baseline, and also at 21 minutes after Ali induction, and we can see that there is clearly more water content in the lungs from when Ali is performed at night than when Ali is performed and in the day. Is viewed here. Here's the graph of this quantification, and it has a correlation with the survival of the mice. They saw that this has an effect, and when you put CT5, when you perform Ali in CT5, you have more survival when this is performed in the night. So upon Ali, lung injury in mice is worse if the inflammation and induction occurs at night. It correlates with more net formation at this time. So we can see that the effects viewed in the proteome and granulation, net formation has a direct impact in the tissue that injury that this neutrophil can cause. So they want to know more. They want to have a more.
Before you continue, I was wondering maybe what's the what happens if you continue this, because we're only seeing 2 hours here in net formation, but maybe these old neutrophils that cannot produce many nets are just recreating young neutrophils, and maybe after some hours, I don't know how fast they could come, they are the net formation reaches a peak. So maybe, I don't know if you the authors tried this, but it could be that it's just the net formation is delayed in this mouse model.
Oh, there's like a second wave of neutrophil reclutation. No?
Mm-hmm. Yeah, that's a possibility. We never tested it. So we did basically 2 hours because that's what we see in the survival experience of the acute lung injury model. Most of the death happens in the first 2 hours. So that's why we chose these 2 hours as the time to look for them. So, but yeah, it's an interesting possibility. We never really checked. But one thing that we know is that in this model, most of the deaths happen in the first 2 hours. So if you leave the mice for longer, there's usually they survive.
Okay, thank you.
Yeah. It also creates some confusion, precisely for the reason that you said, that you don't know if it's a matter of degranulation and nets, which is a very acute event, or it's something else. And we wanted to avoid the something else because then we don't respond to questions. That's another practical question. So in this case, it was a win-win situation. It was a faster model and cleaner. Maybe interesting to look for downstream consequences of all this that we have not looked at in this paper.
I have a question regarding platelets. So you show that your platelets are there's no difference, but you don't see any differences in platelet neutrophil aggregates neither, or platelet activation markers, they are all the same?
Well, we not for this project, for a different project, someone in the lab was looking at these, and there's some diurnal variations, day-night variations in platelet numbers, and maybe quality. They're faint. We don't know what they mean. And neutrophil platelet rates, we also did see some differences, but again, there's a lot of variability just because of the assay that we use to measure these. Yeah, bottom line, we did not go into I mean, that was a project we wanted to work on, but it was so tricky to get reliable readouts. I mean, thing that we're looking here at the spontaneous differences day and night, so that makes it tricky to find reliable markers. It's not like when you induce an inflammation or a strong response where it's very easy to read. And with plateletsnet, we never felt confident enough to you know, we don't have very obvious phenotype.
Okay, thank you.
Thank you, Andres. So we are here. Sorry. Well, to have more evidence of the direct effect of this neutrophil variation profile through the day in lung injury, they evaluate the granule presence in neutrophils that have not expression of Bmall1 and that didn't have the CXCR2 gene. And we can say that Bmall1 depleted neutrophils are more like nighttime neutrophils, and CXCR4 depleted neutrophils are more like daytime neutrophils as CG5. But indeed, there is less, includes less granules in these cells, and we can see that in this graph. And this is interesting, and this is well, this is logic, because if you eliminate the Bmall1 in these cells, you can see that the variation driven by the Bmall and CXCR2 signaling in a circadian manner is low, and they lose always like night-like neutrophils. And if you didn't have CXCR4, you have more an enhanced and non-regulated CXCR2 signaling in these cells, and you can see that the granulation is more present. Any questions?
As a way of this is Andres. So as a way of self-critic here, for us it was very difficult to discriminate in the CXCR4 mutant, which has a very strong phenotype. It's difficult to discriminate between what is that the granules are not formed or that the cells are excessively degranulated. So in this case, because they don't have CXCR4, the neutrophils leave also early the bone marrow. So that's one thing. And they don't control, on the other side, they don't control the aging process, so they overage, like we proposed in the previous paper. So as a self-critic, the reason that they have less granules might be a combination of both. They granulate too much, but they might start off by having less granules. So it's not the perfect model. It's the only model that we had, and it's nice because it matches basically what we see in the wild type mice at different times. But just bear that in mind, and that's an ongoing critic and something that we you know, would like to solve at one point. We just don't have the right model.
Oh, okay. Thanks for the acceleration at this point.
Yep.
Well, they also evaluated this granule presence of these neutrophils by electronic transmission microscopy, and we can see that there is less granules present in the CXCR4 depleted neutrophils than wild type or in Bmall1 depleted neutrophils. Indeed, there is more granules present in these Bmall1 depleted neutrophils. We can see it here, and later. Bmall, they evaluated the net-like structures formation, open alley induction in mice, and we can see that when this might have depleted Bmall one in its neutrophils, there is more net like structured formation, and this is there is the opposite effect in the CXCR4 depleted neutrophils. We can compare that with the wild type conditions in the different daytime, and also they evaluated the edema or the water content in the lungs by computerized tomography, and we can see that is the same type of response. There is an augment in the edema pulmonary in the lung edema when there is no Bmall in the neutrophils. This is reduced, and CXCR4 depleted neutrophils, and also this correlates with the survival of these mice. There is interesting that the authors say that we can say that CXCR4 depleted neutrophils have a protective role in these events in acute lung inflammation. We can see that when there is no CXCR4, the tissue is even have the same edema level like the baseline condition. So this is interesting, and we can see here the process of the intravital. They show a key intravital microscopy of the lungs during ALI, and I put this video to show you that they view that there is a different ALI formation, different types of nets for formation structures. They show that there is flowing nets that are nets that can circulate in the tissue, and also there is sorry, my computer is there is other nets.
The nets are attacking your computer, Emmanuel. The neutrophils are attacking your computer.
Yeah, I don't know why.
Well, while Emmanuel fixes his computer, I got a question. When I was doing some research about this topic, there's some report in 2015 from the Paul Frenet group that he says that neutrophil aging it's dependent on microbiota. So how can we integrate this data with your data that it seems that you conclude that it's an intrinsic process in the mice? So if it's intrinsic, why it's dependent on microbiome?
That's a sensitive question. Jose, you want to answer?
Yeah, so both things could be compatible, because you could have an intrinsic program in the cell that's controlling this, but this is a process that could also be controlled by external cues. And actually, if you take a look at the model that we propose, which is this process being controlled by CXCR2, CXCR4, those are both receptors on the membrane. So these are accessible to external cues as well as internal cues driven by the molecular clock. So both things could be, actually, I think both things could be happening. But in these precise matters, what we did is we tried to check what happens with the microbiota, and we were not able to measure any differences in our hands. So we don't really know we don't really know what's going on there.
Yeah, basically, we don't agree very much with interpretation of that paper. The readout of aging in that paper was not very clear. So there's an effect of the microbiome, but we're not sure that it's in bona fide aging or something else. So that's the answer to that question. It's not. It's basically we don't very much agree with interpretation of it. If you go to the immunity paper, actually, I think there's a figure, a whole figure, trying to reproduce the data from that paper.
Thank you. We'll have to wait a minute.
Sorry, I'm trying to present again. There was a problem, and let me open the presentation again. Sorry.
I got another question. Have you compared the protein content of different neutrophils that migrate to the tissue? Because you mentioned that there's a homeostatic migration towards different tissue. So I'm wondering if the neutrophils found in the lungs or any other organ, the contents of the proteins are different.
We have not checked proteomics on these, but we've checked the granules, and it's actually a figure that Emmanuel showed. So what we checked is that the granule content of neutrophils migrating to spleen, lung, and liver was lower, but we didn't do proteomics.
Okay, thank you.
So
that would have been cool, but it's also difficult. The numbers might be limited in some tissues.
Thank you.
Okay. Well, after that, they want to know if this process and phenomena that they saw in the mice are present in humans too. So they isolate neutrophils from healthy volunteers than healthy volunteers at different times of the day, 8 a.m., 2 p.m., or 7 p.m., and they analyze the hematology, proteomic analysis, granule content, and net forming assays on these neutrophils. So in the about the proteome analysis, you can see in B the results of gene ontology analysis, and we can see that there is some differential enrichment of the process in the proteome of these two type of the neutrophils. Here, I think that is showing the comparison between 8 a.m. and 2 p.m. neutrophils.
Yes.
In those terms, and well, then in C they do the comparison of the proteomics data with RNA sequencing data, and they again don't there is no correlation between these two data sets. So they say that in humans, as in mice, there is no correlation between transcriptional change that impact in the proteome change viewed in these neutrophils. So they also looked for the enrichment of the proteins present in the granules, and we can see that there is more enrichment of these proteins in the neutrophils that were from the daytime at 8 a.m. Well, they put like the nighttime but well, 8 a.m. Versus 2 p.m. Where there is more neutrophils like daylight. And they also analyze the number of neutrophils, and we can see that there is a variation of neutrophils circulating across the blood, and at the daytime we can see an increasement during the day at 2 p.m. or at 7 p.m. Well, this is also there's an effect of the granularity of these cells. We can see that even if there is more neutrophils during after the afternoon, there is these neutrophils are less granular, and we can see here by size scattered measure of these cells, and also they have evaluated number of granules of in presence in T cells, and we can see that also that there is an decrement in this in the granules present of these cells. They evaluated by confocal microscopy. We can see the example of this image here in the right. We can there is a less granule content in the neutrophils in the 7 p.m. and the more granule content is present in the early in the morning. Also, they evaluated the granule content by transmission electron microscopy, and we can see indeed less density dots, electron density dots or granules presence in these cells in the afternoon. And if they are when they analyze the proteome of these two type of cells, we can see that differential enrichment of different type of proteins in these two data sets. So it's more clearly indeed that when you can they analyze for the proteins present in granules, there is a more enrichment of these proteins in the early morning neutrophils than in the afternoon neutrophils. And well, this recapitulates the effect that are present in mice. So after they evaluate also the ex vivo net formation capacity of these neutrophils at a different daytime, and when you these neutrophils are from patients at early in the morning at 8 a.m., they show that there is net formation, and this is diminished in the 14 hours, and in the 19 hours it is also there is again increased in some reason. And well, they also do an interesting insights into how this process could be impacting in the pathology progress of or in the clinical signs viewed in patients with pneumonia. They measure pneumonia severity index by retrospective analysis of patients. And we can see that this analysis indicates that they analyze it by these patients by the time of the day that they were ingressed they become on the unity of care, intensive care unity. So we can see that when they ingress out this unit, this unity at the night, they have more severity pneumonia index than when they are in the ingress in they are in this ingress to this unity of the daytime. So there is an effect of on the death. They analyze and they show that there is a correlation. There is a variation of the death frequency across the daytime, the time of the day that there are when they enter in the care unit. So this indicates that maybe the process the process of neutrophil disarming and the neutrophil program proteome and capacity of net formation and the granulation across the day, change across the day can include maybe impact the severity of the damage in the inflammation in the inflamed tissues like the lungs where neutrophils arrive. So finally, the model of this study, they propose that when neutrophils are released from bone marrow after its granulopoiesis, they are released like fresh neutrophils. That well, they proposed that in the last paper, and then when neutrophils are in the circulation, they drive a diurnal proteome change that impacts the granulation pattern and also the net forming capacity. So they propose that this is due CXCR2 and CXCR2 role, where the circadian clock by Bmall is controlling this axis of signaling, and this promotes the neutrophil disarming program. So they also propose that CXCR4 are inhibiting this process. So there is two panorama. When you can have more net formation, more vascular damage, and less survival when the neutrophils are not driving this disarming program. But if the neutrophils arrive with this disarming program, you can see less net formation, less vascular damage, and more survival. So we can see that it is a mechanism that permits that neutrophils that arrive to the tissue, to the lung, and specifically, can't cause many damage as they can cause if they were arrived like the pattern when they are released. So this is the final figure of this paper. If there is any questions, please say it. Thanks.
Thank you, Manuel.
So we can see now we are going to the questions asked to the outer section of this. Let me.
Okay, I have a question. When neutrophils lose their granules, the content of these granules is released to the circulation?
So that's a good question. The answer it's we don't know, but we have some hints because we analyzed in this figure where we analyzed neutrophil elastase activity in plasma. So what we saw there was that as neutrophils were releasing the granules, the elastase activity in plasma was increasing. So at least it seems that neutrophil elastase may be released to the plasma, but we only know about that.
Have you, like following up that question, have you tried other, like maybe the proteasome pathway or maybe autophagy that can change the protein number inside the neutrophil? Have you analyzed if there are changes on that?
No.
That's a good point.
Yeah,
I'm thinking that autophagy was involved a couple of years ago in maturation of neutrophils, but we have not looked in circulating cells.
Yeah, I was like thinking about that paper too, about how it's about maturation, and maybe the autophagy is also playing a role in this continued maturation of the neutrophil.
Well, you have to think one big difference is that all these things occur relatively fast within 2 or 3 hours at a time. The process is relatively fast. For autophagy or more cellular processes, I'd say it's hard to compare because we don't know, but some of these things might take longer times. In particular, in these developmental, in this paper showing a role in the development of neutrophils, that's many hours or days even that the process might be active in the progenitors. So yeah, it's hard to compare. We don't know.
I see. Thank you.
And thinking about the destiny of those content of those granule content, it must be it could be a a mechanism that control that all that enzymes that are potentially damaging the organs? Because if neutrophils release the content to the blood, thinking about humans, neutrophils are a higher subset, the highest subset of leukocytes circulating through the blood. There could be really high amounts of those enzymes in blood, and it could be a mechanism that control that these enzymes didn't or do not cause damage to, I don't know, the endothelium or something like that. Because for me, it's very difficult to imagine that it could release through the blood, and it doesn't exist a mechanism that inactivates of all these enzymes. What do you think about it?
That's a good point. So we have a reviewer that was a little bit along the same lines as you were, and he wanted to see whether there would be also accumulation of these proteases in tissues that could cause damage. So we measured that, and we did not find, if I remember correctly, Jose, we did not find evidence of elastase. And the reason we did elastase because it was relatively easy to measure, and we could measure also activity, not only protein. So we just tried to keep things simple. The other consideration is that the release, if you look at the curves that we saw, elastase is relatively slow. At one point, the levels are high, but you have to think that that would be in the whole soup of the blood rather than concentrated on a specific area, which is what you would get when you have an acute response. A lot of millions of T cells acting on the same spot, so that might be damaging. And another consideration is, well, you know, that's the typically we have these conceptions that come from disease models where all these molecules are damaging, and that might be the case or that might not be the case. Some of these molecules, and elastase is one of them, have been shown to be also able to do remodeling or to even do signaling. So we don't know if some of these enzymes, proteases, but also some of the antimicrobial peptides that are released might be having protective or even signaling functions. This is all the preconception that elastase is bad, myeloperoxidase is bad, proteinase G is bad. It might not be the case. I think that's in general, this with neutrophils and in general in immunity, there's a lot of preconceptions based on very specific examples of disease, and I don't think we need to be. We should be a little bit less. What's the word? Categoric, less. I forgot the word. It doesn't come to my mind now. We should be more relaxed about the data, or about the interpretation of the data. A protease might not necessarily be damaging when it's in the plasma and when it reaches healthy cells. Basically, that's what I mean to say.
Yeah, I agree, and also there's in plasma there's a lot of protease inhibitors that would be acting, I imagine.
Okay, thank you.
Hi. Well, I have a question about this work, and well, there is known that many molecular clocks or many clocks present in different tissues of cell populations can be coordinated between them and with the central clock in the suprachiasmatic nuclei. So I want to know if you know if there is a communication or coordination between the molecular clock of autonomous in the neutrophils with another molecular clocks present, like the central clock in the suprachiasmatic nuclei, or in another, or with the clock present in another tissues. Do you know if there is coordination or communication?
That's an interesting question. How much more time do you have for an answer? I'll just to be brief. So the suprachiasmatic nucleus mostly would regulate these by producing inducing the production of hormones like glucocorticoids, for example, which we did not explore in our paper. And the cell has an autonomous clock. How do you put the two things together? We don't know because we have not interfered one with the other. What we've done is to show, and this is experiments that we're doing now, that if you only eliminate the intrinsic clock, you get effects not on numbers of the cells, not on the oscillation. You get an effect on the effect and in the inflammatory response or the infectious response. That we know. So one possibility is that one controls numbers and trafficking, and the other one regulates intrinsic properties like the granulation or transcription of a specific proteins that they will need. We have models now where we're trying to address this in a little bit more systematic way, where we have the full knockout mouse with the suprachiasmatic nucleus. So in general, the systemic circadian response is gone. The mouse is arrhythmic, and so is the neutrophil, and we will see if this mouse is completely messed up, which is what we expect. It loses the day-night changes in inflammation or infection, and then just made one in which we rescue specifically Bmall only on neutrophils. So now we have the three conditions, or the four conditions. Everything has its own clock. Nothing has a clock. Only the parenchyma, but not the neutrophil, has a clock, and only the neutrophil, but not the parenchyma, has a clock. The parenchyma, the brain, and perhaps we can answer these questions. I'm guessing that there's a communication. I suspect that it's a matter of different tasks. One regulating more migration and numbers, the other one regulating activity of the cell.
Okay, thank you.
And also, just as a brief comment, so we know that there's as Andres was saying, there's a connection between the two, the internal control of in the neutrophil itself, and the external control by the light or any other cue. But what is the precise link? We don't know. There's one thing that I think that's interesting, that CXCR4 is involved in all this process, and we know from the previous paper that the ligand CXCL12 in plasma is fluctuating in a circadian manner that coordinates with the aging state of the neutrophil. So there's many things that we don't know about how these two systems correlate, and that's going to be very interesting to know. I agree.
Yeah, sure. And also, it's interesting how in the steady state, neutrophils are released from bone marrow in a specific hour in the night. So it could be maybe CXCR4 role in this induction of release. I don't know. You know how is this releasing from bone marrow in a specific hour? How occurs?
How that happens? Well, that's been studied more for stem cells than for neutrophils. And in that case, the simplistic conclusion was that the levels of CXCL12 change during the day. So if you look at the transcript of protein level, there's an oscillation up, down, that would coincide lower levels of CXCL12 in the bone marrow, more release of stem cells or neutrophils. That's the simplistic idea. How this is regulated, so one piece of evidence is that there's direct innervation of the cells that produce CXCL12 in the bone marrow. So when you get signals directly from the nerves, in this case, they're sympathetic signals like adrenaline or noradrenaline that hits those cells and suppresses CXCL12 expression. That's a complicated way to suggest that this is regulated. And that's very interesting also because it establishes a connection between the brain, stress, systemic inflammation, and regulation of the signals that hold the cells in the bone marrow or make them come out. More stress, more inflammation, more in general damage to the host. So there's a lot of people interested in this particular loop of regulation of the release of stem cells or neutrophils or anything from the bone marrow.
Well, thank you for your answer.
I have another question with the trying to connect with the last one. So you don't have any idea if the precursor of the neutrophil function is driven by circadian clock? And my second question will be, I will try to make myself understood. It's kind of complicated for me to explain, but you show all of this in an homeostatic state. My question will be, what would happen in inflammation? If in inflammation if there's any inflammatory signal that induce a loss of this circadian intrinsic function of the neutrophils.
Okay, so the first question.
I think Aranzazu, you don't sound like an Aranzazu. Yeah, it must have been a review. The second question, to start with that one, there's one that was raised at one point during the review of the paper. If inflammation alters this, and when we did an experiment where we used inflammation, then look for the recovery of the signal. It came. There was a transient elevation in neutrophil counts and phenotype, but as soon I think it was 1 or 2 days, Jose Maria, that the system went back to normal. So the recovery was very fast, at least with the resolution that we could measure. What we hint from the models that we have studied is that inflammation changes the amplitude of the response, but not the rhythmicity. I don't know if that makes sense.
Okay. Yes. Thank you.
And the first question, I haven't understood either.
If there's any, if the precursor of the neutrophil has also a rhythmic clock, the precursor.
Okay.
Because we also have this in the neutrophil, in the developed neutrophil or the mature neutrophil, but what happened if in the precursor?
Yeah. Jose?
The answer in my case is I don't know. So we never looked into that. Circadian and the neutrophil precursors, which I don't know.
Your guess is that
someone in the lab
is dependent on.
Someone in the lab has been looking at changes, not with the central solution as we've looked at neutrophils, but changes in numbers of different progenitors, and there's some changes in some progenitors, but we don't know if that's regulated by the clock. That's a different project, but we're not looking so much into the clock of those particular cells, but the clock of the bone marrow niche. So it's different. We're looking more at the regulators than at the leukocytes themselves. We haven't looked. One interesting thing that we were always trying with in our heads it's interesting that CXCR4 keeps the neutrophils in the bone marrow, but it's also a negative regulator of the clock. So the idea that that I like to play with is that as soon as a neutrophil leaves the bone marrow and it's away from CXCL12, so the agonist, then the clock starts running because the repressor of the circadian clock is lost. The signal is dominant in the bone marrow. So like the bone marrow is a tissue that freezes, that blocks by default the circadian clock. That's an interesting, conceptually, it's an interesting idea.
Thank you.
Can I ask a question? So instead, I wanted to ask if you think that from also this work, you may manage to, knowing so well now the proteome of the neutrophils, you may manage to find those proteins that are somehow necessary for the neutrophil clearance when they get to the bone marrow.
Jose, do you want to answer?
So the question was if we can find with the proteomics the proteins which are necessary for the clearance in the bone marrow.
Yeah, because I think it was also your 2013 paper where you showed actually that at a certain time the neutrophils get to the bone marrow and macrophages are involved in their clearance, but then actually, of course, the molecular details there are very tough to to understand. So I was thinking if you think it's something interesting, or actually it's just a matter of philosophy here.
No, it's an interesting thing. We don't know much about what's driving that. We know that the neutrophils have to pass through this process of aging in order to go in homeostatic conditions to the tissue. We know that. Certainly, we could try to analyze this the proteomic data set in more detail to try to find what's driving that. We didn't do that. I don't know if we will do it.
Yeah. Well, one problem, I mean, proteomic is cool. The problem is that you can make a claim of the proteins that you see, but you cannot.
I lost Andres.
Andres, we're losing you.
Sorry, who was talking? Who was speaking?
Hey, Andres, I lost you before because of the connection.
Andres? Yes. Oh, Andres is not connected.
Okay, never mind. I mean, we can go on then. Thanks for answering, Jose. Anyway, thanks.
Thank you, Jose. Well, I have any question. Later if Andres gets in again, we can retake the question of Federico. But well, Andres, I don't understand what implications can be said about the data that you obtained of patients with pneumonia. Do you say that you show that there is a correlation in the there is a direct effect of the hour that these patients are entering the intense care unity with the prognosis of its pathology on the severity of the pneumonia. So, what do you can interpret of this data? I don't know what exactly means that that determined time a patient enters to the intensive care unit. Can you say any of some of these?
Well, there's one thing that we've been seeing in many of these models that we've been using, sorry, at the different time points of the day, is that the moment when the when the disease starts, it's very important for the outcome, even days in the future. So that's something that's very interesting. And in this precise case, I'm sorry.
Don't worry.
In this precise case, what we did is analyze the correlation between the time when the patient goes to the ICU with the outcome in the end, right? So as we saw that in humans, neutrophils also do this disarming process. And we showed that early in the morning, neutrophils in humans have more propensity to form nets, and they have more granules, and so on and so forth. We wanted to check what happens between so if there was a correlation between these net formation, increased net formation, increased number of granules in human neutrophils, and the outcome of the pneumonia disease. It was a way to try to check in human patients something as similar as we could to the acute lung injury model that we were using in the mice.
Okay, thank you. Do you think that these different responses by the daytime that you presents the lung inflammation, is have a role in the heterogeneity of the other responses, immunity responses, actually in the COVID patients, in the COVID-19 patients, or in the different prognoses that many people have acute lung injury and have deficiency in the respiratory capacity.
It could be, and it has been proposed that nets actually may be playing a role there as well. So there are things to be looking at, I think. Yeah.
Okay, thank you.
Sorry, about nets again. What does change between the adherent and then instead the non-adherent ones, the ones that go with the flow, let's say? But because like I read also a paper about the intervention of neutrophils in NETosis in malaria, for example. I think it was from the Zikninsky lab, and there it seems there to be also a connection with sequestration. So like maybe a density of adherence protein on the activated endothelium. Do you think that this is normally the case actually in general? And do you think that this actually makes sense because it improves the defense? Because it let's say concentrate the nets there where they are needed, so where the endothelium shows the most inflammation. What's your thought about that?
Yeah, so in our case, this came as an observation. We were looking at the images, we were looking at the nets being formed, and one of the things that struck us was that actually in this lung injury model, most of the nets that were forming in the lungs were, we call them flowing. So these are the nets that you see the neutrophil, you see the net starting to crawl out, and it's released and it goes with the flow. It goes somewhere else actually. And then there was this other type that we could see where the neutrophils would stick the net to the endothelium, and it was an active process. I think that we don't know, but I would guess it was an active process because the neutrophil would crawl on the endothelial surface and would leave the net behind as it was crawling.
Wow.
Yeah. So it's two very different ways of forming the nets. And actually, this is one thing when we then try to quantify nets in the tissue. This is something I from that time on I try to keep in mind, because if, as I saw in this particular example, most of the nets that were forming were actually these flowing nets. You basically lose them with the flow. So when we take then the sample and analyze it by microscopy, we may be losing most of the nets which are produced, if this is the case in all the tissue, which we don't know.
It's also interesting that people are using more and more nets in plasma as a marker of certain diseases, but of course it might be more relevant those that you don't see in plasma, those that remain attached to the vasculature, because those would be the most toxic.
Okay. And just this, do you think that nets does mean the end of neutrophils? Or like does it mean like it's a condemned to death for the neutrophils, or actually they like it depends by the amount of nets released? Like if it is, for example, just mitochondrial DNA, then okay, then and the neutrophils is fine, or otherwise, if it is the DNA, the host DNA, then this means death automatically.
I think only Paul Hughes has made the observation that nets and the neutrophils might continue crawling and eating pathogens in a specific context of skin infection. In general, what we see is that the cells seem to be still moving, but we don't know what the cells, after they release the DNA, when Jose images those in the lung and then they spit out this thing. For a while, they seem to be moving. It's not like they stop and disintegrate. So what that means, it's unclear whether those cells that have lost the nuclear DNA are still capable of damage, or what's with them. That's actually something we don't know. That is very intriguing that there might be many more dead cells or cells without a nucleus running around in the tissues or in the circulation. We don't know, and that's why before I was saying let's try to be less dogmatic, because we know what we know, but we have no clue of what we don't know. Always, even if it sounds a little bit redundant.
Okay, that's very interesting. Thank you very much.
Guys, I'm gonna have to leave because I have a little one that demands my care. So I just wanted to, you can continue, and Jose is here, and actually Alejandra, I think, joined, he's also the second author. But I wanted to thank for this super cool initiative. I'll try to join for Arturo's and for Sarah's presentations too, if I can. So yeah, thanks very much. Very cool. Gracias.
Thank you very much. Muchas gracias por estar aquí.
Nada, placer.
Jose, I have another question. What drives the net production or release in the tissues under homeostatic condition? Because I was wondering that this could lead to a constant inflammation signal in the tissue that finally could lead to damage, to tissue damage.
Yeah, so well, I'm not aware of anyone that's shown that nets are released in the steady state. So if that's the question, I've never seen nets in the steady state. So nets are always, as far as I know, a result of some inflammatory insult. So in the steady state, I don't know. I would not be very surprised if there was a role for nets in the steady state, because I don't know. But yeah, so in the steady state, I'm not aware of nets being formed. I don't know if your question went on that line.
Thank you. I think I had my confusion of that, but thank you.
Okay.
So I don't know if you guys have any other question. No. Well.
Okay.
Yes. Can you hear me?
Yes, yes.
Hello?
Yep. Yeah, we can.
Can you hear me?
Yep.
Yep.
Yeah. Okay. So I just wondering two big questions. One is, I was wondering if you know how protein disarming could be evolved to improve neutrophil activity only during night.
That's a good question, and one that we have thought actually about a lot in the lab. So one thing that we think about, so the whole system, both the whole system seems to make neutrophils during the night more active, more prone to form nets. They have more granules. They are more ready. They can more readily respond to injury, to inflammatory signals, and so on and so forth. So that's a good question. So why would we want neutrophils at night being more reactive, actually? So we don't. The answer is we don't know. We know that this is controlled by the light through the retinal ganglion cells and so on. But we don't know why would we want neutrophils at night being more active. One thing that you can think of is that during nighttime is the active phase of the mice. So if neutrophils are more prone to defend the neutrophils during the night, then the mice would be more protected during the night. That's one of the ideas. But the quick answer is we don't know.
Okay. So I have another question. Are you planning to maybe characterize the metabolic profile that is shaping the shortness of life of neutrophils and the net formation?
The metabolic profile. So I don't know if Andres should be here to answer because I actually left the lab.
Okay.
So I am not, and I don't know if he's actually going to look into that.
Okay. So thank you.
Okay, so I don't know if you guys have any more questions. I would like you, if you don't mind, if you could turn your camera so we could take a picture of the meeting. Okay, there. Guadalupe. Usted, alguien más que tome también, porque si no yo no salgo. A ver. Okay. Once again. ¿Va otra?
¿Sale la mayoría?
Sí, salen todos.
Ok.
Pues muchísimas gracias, José. De verdad que estamos muy agradecidos por esta oportunidad y que te hayas tomado el tiempo de estar con nosotros.
I have a quickly question. Jose, can you say what are you doing now in the Micala lab in Cold Spring? I interested in what are you doing now?
So right now, so I came here. I joined the lab to start doing cancer research. But with this, so I just arrived when this virus situation was just unfolding. So right now everything is like halted, and what I'm doing actually now is trying to apply all these things that I know of lung injury and so on, and like things that you've seen in the paper, to try to apply that to the COVID-19 situation. That's like everyone else. So.
Well. Okay, interesting. Thank you for coming, and thank you for accepting the invitation.
Thank you. It was my pleasure. And just one thing, if there's only one thing you take from this presentation is things change in circadian manner. Take that into account when you plan your experiments. Don't do some experiments during the morning, some experiments in the night, things like that.
Yeah.
Things change quite a lot, quite dramatically. That's a take-home message, and I think the most important one.
Yes, it's a source of variation.
Yeah.
Thank you.
Thank you. Thank you guys for joining us.
Thank you so much.
Thank you all.
Bye-bye.
Thanks. Bye.
Bye.
Súper informal la despedida, amigos.
Porque no sé